to present. Yes, but essentially what I want to transmit to you, that anything that you could stain and you could see under fluorescent light, we could capture with a confocal microscope. Okay, anything, even if it goes like that. Okay. I'm from the Primate Center of Tulane. It's very near from here. It's an hour in Covington. Okay. Let me see. With this one. Okay. And what I'm going to talk today is about confocal and what do we do with confocal, and trying to give you some ideas of what you could do. And if you want to come and work with me, you are welcome. I'm in Covington. You need to contact me or ask Mark where we are, and then we could establish some collaboration. Okay. Confocal microscopy is the best microscopy that you could get with light. As an example, I have this. This is a vessel. Okay, all this. Oh, come on. What I did? What I done? This is a vessel. Okay, this is a vessel. Okay, and you have never seen a vessel like this. This is inside the brain. This is a vessel going like this. Blood flows internally. These are some cells that are outside of the vessel. They have extravasate. They are moving around cells. This is outside. This is outside, and this cell is coming out. Okay. We tag the cells with different fluorochromes in order to follow them. This particular color is, we are giving them to tag monocytes. Then these are monocytes coming out of the vessel in the brain in order to go to the sites of an inflammatory process of a SIV, of the simian immunodeficiency virus. Okay, but this is the kind of resolution that you could get with a confocal. The stack was of about 70 microns. I had problems with the movies, but if I show you the movie later on, you will see that we could do lots of movies. Okay. Okay. Then I'm the manager there in the Primate Center, and my main research is in neuroaids. So the person that asked about the something to stain about Alzheimer's disease, we had a project with someone from Lady Davis in Canada about the detection of plaques in the brain. And we don't use an antibody, so we use thiosulfate. It's reported in the literature. Thiosulfate, and it stains the plaques of Alzheimer's, and it's very good. If you want, I got some pictures there, but we'll see at the end. Okay. All right. Then for you, my position will be to try to help you to gather the best picture that you could get, yes, in order to show what you want to show. Okay. A lot of times, the best advice that I could give you is how to stain the thing, yes, and then how to collect it. That is the confocal microscope that we have. Is SP2. Is when I tell everybody it's an old one, I want a new one. We have a grant application for the new confocal microscope. Okay. After November, I could tell you if we have a new one. Okay, then confocal microscopy is the best microscopy, light microscopy that you could get. Is the one that is going to give you the highest resolution. Okay, and then we could penetrate in tissues, inside the tissues, in order to gather the light that we are going to use to reconstruct whatever we see there. Okay. it allows you to do multi-dimension imaging, and with this, uh, um, sometimes people say, what are you talking about these dimensions? But it's the depth, the length, the width, time. Okay, we have 4 there. Then if we have each color as a dimension, then we have 4 dimensions for the first color. Then when you have another color, you have 5 dimensions, and so on. Okay, mathematically you could have n dimensions. Okay, then that's when I start talking about doing equations in different dimensions. The resolution improvement is because we get a way, we throw away all the light that is not perfectly resolved with the piece of equipment that we use. The contrast improvement is because we use we discriminate between the focal planes. That is the basic. I will touch that later on. And we do optical sectioning. If you understand what is the optical sectioning, then you know exactly what the confocal microscope is doing, and I will try to explain. Okay. And nowadays we could do pictures with many colors. Normally in our facility for 3 colors, with planning very well 6 colors. Okay. With the new microscope, I hope that we could do at least 8 and probably 10. Okay. Oh, I'm going back. Sorry. Okay. Then the basis for the confocal microscopy. We use fluorochromes always. Okay, and it's a fluorochrome that is different than for flow. The fluorochromes that we use are the fluorochromes that give you the energy slowly. Okay, the ones that we use a lot are the Alexas, and I will tell you. Then with the spectrum, we excite it here, and we receive the light here. Okay, this is the excitation, and this is the emission. These fluorochromes are all, and then they cross over between one and the other. Okay, the new generations are the Alexas. Okay, but these bases are the fluorescent bases of the confocal microscope are the same fluorescent bases for the the cell sorter. Then we have the same things, and sometimes with some of the fluorochromes, we could excite with one line and receive the signal in 2 different places. It depends on where it fluoresces. Sometimes some substance fluoresces, we excite it with a blue laser and fluoresce in green, and some of them fluoresce in red. Okay. Then with less lasers, we could have more colors. The generation. The Alexas are considered about the 4 generations of fluorochromes. FITC, TRITC, they are the first ones, and they are very good. And if you don't have any other, that's okay. You use them. But normally, when you want to do more, you need to use Alexas or other modern fluorochromes, like Q dots, that they are nanocrystals, and then they have better defined spectra. That's the reason that we use them. Okay. Then the confocal principle is anybody has burned paper with a magnifier? Yes, no? Had you burned paper? None of you have burned paper? You are very good. I can't believe that you did not find a magnifier and burn paper. Make fire with a magnifier.
Works in the desert really well.
Here too, any place. Okay, when you are burning the paper and you move your magnifier up and down, you reach the point that you have a little dot, very bright, and then the paper goes. When you find that point, you are finding the focal plane of the magnifier. Okay, that is the focal plane where it will focus all the light in one little dot. That is the focus of that magnifier. Just imagine if light comes through the other side, the side of the paper, then you have the paper to the other side, there will be in exactly opposite direction at the exact distance another point that will focus the light from here to there. Okay, I'm going to include that slide next time. Burning paper. Okay, and then the relation of these two focus to the magnifier is that they are confocal, and that's the beauty. You see one point here, and you see another point here only. Okay, with that, we see only one. We shine the light here. Okay, we see one. We illuminate one point here, and then we receive here one point only. Then, when we switch on the laser, we see one point. Okay, and then I will go one point and say, oh, what I'm going to do with that? Probably nothing. Okay, if I move the laser, I have another point, another point, another point. Okay, this is supposed to be a straight line. Okay. Then we have a line. That is the scanner capability of the confocal. You go line by line, okay? And you have a collection of lines. You have, when you finish, you have a plane. 2 lines at least have a plane. Who knows what defines a plane for an optical cut? What's a plane? No? In mathematics, what defines a line?
2 points define the line, 3 points define the plane.
Very good. Hey, you could work with me. Exactly, 3 points define a line. A plane, sorry, a plane. Good. Okay, then with the microscope that we have, it's designed like this. Leica, from my point of view, is the best one because it has a spectrophotometer. It's the best one for a multi-user, for a core facility. Okay, but because it has a spectrophotometer. Then we shine the light of the laser, goes here, goes down, excites the fluorochrome, comes back, and then we separate the light there. And we do that for 3 different lasers that we have, and then we form an image. Then the optical slicing concept that the confocal microscope does is that by scanning line and then a plane, it has a very, very, very narrow cut of whatever you are imaging. Imagine that this is a cell, and this is your lens. It's going to shine, and then we move it around, and then this way too. Then we have one plane, and we have here, and we do this, and we go down on this. Okay. Then when we finish, we could separate the slices, and we have them like this: the top of the cell, something inside, the nucleus of the cell, the nucleus larger, some organelles, some lines. Okay. Then we are doing optical slicing. Okay, and then with that optical slicing, we could do at least four dimensions. Okay, X, Y, and against time as 3 dimensions, and that's a movie. All these movies that we do digitally is that just X, Y, and against another dimension, time. Had you ever done a flipper book when you are bored in school that you draw at the age of 1 and you go like this? Yes? That's the basis of making movies electronically. It's exactly the same, one image after the other. Okay? And if we put another dimension, then we will have the 4th dimension. Then we could do volumes against time. Okay. This microscope that we have is equipped with a differential interference contrast. And this is something that is very interesting. It was invented about 200 years ago. very but it is the capability of seeing things in tissues or in cells without staining. And it uses two polarized filters. And you come with me to the place that I work with. I will show you two polarizers and you will understand perfectly. The polarization of the light is just that if this vibrating in many ways, the light, the polarizers allows the passage of the light that is vibrating only in one direction. Okay. If we put two polarizers in opposite directions, perpendicular, after two, you have no light coming through the things. Then anything that you put in between will interfere with the pattern, and that's what you see. When you have them in your hands, oh, it's true. Okay, and that is a DIC picture. And yes, I could see some cell here. Oh, yes, it looks like cell because Javier says this is a structure, who knows what. There's something else here. There's another cell here, okay? And that's the piece of meat that we have, or piece of tissue that we have. Then with the confocal microscope, is it going to work? No. No, isn't it? Oh, I could. I need it red. Wow.
The pen is just a prop.
Yes.
You have to pick up the pen though for it to know which one to draw with. Okay.
Then we are not having. I am having problems with the movies, but at the end I could show you the movies. Okay. There was an image of the slicing. Okay. And the slicing was this one picture, the next picture, another picture. In that piece of tissue that I showed you, that it was all these pictures. Then the morphology of the cells looking in their own environment is different of whatever you had seen on the Petri dish or in the books. This is a leukocyte. This is a leukocyte that is moving, and I have another movie hopefully later on, but you could see these are the pods that a cell that is moving is doing like this and pulling itself like this. Yes? But when had you seen this in the real thing happening? You see these pods. Then when we reconstruct all this gallery, the gallery is just a collection of pictures. We could see three-dimensionally how the cell is moving and how is it doing. All right, it was there, the movie. It's not coming, is it? No. Okay. Doesn't want to come. Oh, but anyway, if the movie doesn't work, you could always project all the information that you have into two dimensions again. Okay, and then we have in that piece of tissue, we have a cell that is, and with the movie is more impressive because you could see where this thing is inside the cell. What is that feature in the cell?
Nucleus.
Very good. Who said that? Who said that? You? Who said that it was the nucleus? Very good.
It's June.
Yes, that's the nucleus. This is the tail of the cell. These are the pods. Okay. I have a project where I study cell movement in the tissues, cell movement towards the infectious agent that we have. This is then it's very easy when you see these kind of pictures to say where the cells are going, because the biggest thing that they have inside is the nucleus. Then you could see that these guys are going to make the effort to bring something. They are pulling, okay? And this is without just trailing. Then that's the way that cells move in a tissue that is full of other things. They have to make room, okay? Could you imagine like in the pit in the in the McDonald's, in the pit with the balls? When you go, you have to move things. Okay, it's like that. Okay, they move like that.
Oh, the ball pit.
That's very good, but that's the way that cells move. And this cell is infected with the SIV virus. This cell was going to the point of where we had the virus in the monkey, yes, and it got infected there. Okay, but this is a very good way of studying locomotion. Okay, and if I don't reach that, but at least is to give you some ideas also. How the cells move is important, and then with confocal microscopy, we could do this in tissue, either alive or in tissue from necropsy, that they are frozen in time. Okay, then one of the things that a lot of cells and a lot of organisms go to, we call it chemoattractants, and a lot of the chemoattractants are cytokines. Okay, then I have a project where we see cells synthesizing different chemokines, and we see cells polarize towards that. This is new. It's not published. Will be published probably next year or at the end of this year. All right, something that comes out with a confocal microscope is what is the resolution that we get. Okay. And the resolution, we define the resolution in a confocal microscope different than the normal microscopy, that is just the separation of 2 points. Here is very, very important, all the lengths, all the things and so on, but the critical part is the wavelength of the laser that we are using. As we are very, very near to the resolution of the light that we are using, and also to the resolution of the diffraction factors that we have passing from glass to tissue to glass. Okay, that's the limitant. Oh, someone found me here. Sorry. I didn't switch it off.
Is it going to blow up, Javier?
No. Probably do. Okay. The next one. Oh, and because it's very well defined what we see through different glasses. When we see through glass, we always see this feature, something very bright in the center, and then a halo. Okay, and that has a mathematical function, and with that mathematical function, lots of programs to clean things of the confocal microscope or other microscopes use this Airy pattern as the point to do this. That I want to have all the light that is coming from that point. Here I go here and then here. Okay, and in 3 dimensions, the point spread function looks funny. I'm not going to show you. Then the resolution changes with the wavelength. We have more resolution towards the UV light than in the far red. Okay, essentially only the trained eye and only with making measurements you could see that is different. Okay, essentially everybody sees that these things. Oh, it's the same, but no, it's different. Okay. I didn't know to whom I was going to talk. Then I say, and that's why I included this. Okay. I like a lot the microscope that I have, and it's very expensive, and I have been using it a lot. But there are some others nowadays. Okay. Then this one that we have is the best for a large group, because we could do any fluorochrome that you could imagine in the visible. Okay. But for a little more resolution, you have multiphoton, okay? When you are doing confocal illumination, that's the benefit. Then you don't quench the rest of the things, and then you increase in the resolution, but very little, okay? The new is called the 4P, or the 4 pi, and it is mainly related that it could penetrate more into tissues. Okay, that's one of the things. I think that if we had more users in our center that require this, we will be able to get one, because we use a lot the microscopes. But at the moment, we don't have anything that requires that kind of penetration into the tissue, the order of 1 millimeter or 2 millimeters. Okay, then that's one of the things. And the other is that this spinning disc, the Yokohama way of doing confocal microscopes, and it's an excellent piece of equipment for culture cells. It doesn't have any penetration in tissues, but for live microscopy, you could see organelles moving inside cells. Yes? It's a marvelous thing. Okay, but for example, the 4P is around $500,000. The spinning disc is about 300,000, yes, or 200,000. Then not a lot of people will buy one and say, okay, give me one. Okay. And we have all these series of the convolution microscopes where it's not confocal. What they do is they vibrate either the sample or the lens in order to make to defocus the subject that you are looking, and then mathematically you assign all the light to the respective place. Okay, and they are very good, but it takes a lot of time. It takes a lot of time to get the picture, one. And the other is, I think that it has better resolution through confocal than the convolution. This whenever you find another person that uses the convolution, they are the best thing. The convolution had at the moment a limit number of fluorochromes that they could use, because it's very, very difficult to deconvolve multiple signals at the same time. Okay, but then that will be an advantage of having a true confocal microscope. Okay, then the color formation of these of the computers on the images in a computer, all of them are based in the convention that we had before for the monitors, that they were red, green, and blue. Okay, and with these 3 layers, we could generate millions of colors. Okay, a picture in a computer is like an Excel thing that you have the location and the magnitude. Okay, that's a picture. You have location and magnitude. That's a picture electronically. With 3 of them that we could put together, we could generate colors. If we have only 2, then we could yellow these colors. And with the 3, we could generate all the million of colors that you've seen in your computer. Okay. The reason I put this is that sometimes people say, but from where are you getting the numbers of this? Every single picture that we capture has numbers, and you could do things with them, like counting pixels, like counting cells, okay, like doing associations with one with the other, okay. All right. One of the reasons that we have been successful doing imaging for all this time is that very soon we learned that we could combine different techniques in order to render whatever we want. Okay, there is not an impediment of only using stains, only using antibodies. Yes? We could use a series of things that will fluoresce, and then we could combine them and find out how do they look and if they relate to whatever we want to show. Okay, and a lot of times we find new things with this. just imagine this is the piece of the piece of meat, the piece of this is brain. Sorry, a piece of brain, and yes, we could see with this. This is a vessel. Okay, it's a red blood cell there. Okay, and there is something here that I don't know what it is. Some other, here's a cell, another cell, another cell. Okay, but when we use fluorochromes, we could use a molecular technique to detect the specific virus by doing in situ hybridization and detecting the product of in situ hybridization. It's a molecular technique that is very sensitive. Okay, it's RNA hybridization to RNA. Is very, very selective. Okay, then that's the same piece of tissue, but I can see that that area that I was showing before, it has cells that are infected. Okay, we could use an antibody that has label already. Most of the times it's the most expensive way of doing this. But if we don't have, or we have many colors that we had to put, we don't have one that will do the job, that's okay. And if we have the money, we could use this a lot. Every time that I discuss with a principal investigator, okay, we finish with this, that it's more expensive not to have results than the antibody. Okay, then there's always ways of finding the money to buy these things. Then this is one that is going to detect the astrocytes. This is against protein that the astrocytes produce a lot. Okay, and this one is an indirect method where you use an antibody that is not labeled, the first antibody, and then a secondary antibody. Okay, and then we detect the area where the apical side of the vessel is. Okay, and then we add some stain. This time is ToPro 3 that fluoresces in the far red. A lot of people use DAPI. They say, oh, DAPI, but that fluoresces towards UV light. Okay, then we could put the image all together, and then it's very clear that this is a vessel. We have the vessel here. There are cells that are infected here. Okay, I know what kind of are they, but okay, these are infected, and then all this is what we call a perivascular cuff, like in your hand. You see a large accumulation of cells. This is an inflammatory process in the brain of an animal, and it has all these cells that are infected. Okay, then that tells you a lot what is, then now you could start asking what kind of cells are those. We have been doing that, and those are macrophages of certain lineage that are mature macrophages. The young macrophages or a different lineage don't stain for that. Okay, then there are lots of things to do. One of the things that I'm doing is how these cells came across like this. What do they do in order to go there? Okay, you could study extravasation of cells into the place of the inflammatory process, or you could ask the question that is difficult. Are these cells going this way? Are they coming out again? Is this cell that is infected is going to stay there, or is going to go somewhere else, or is protected and become a big reservoir of virus? Yes? There was someone asking? Were you asking?
Oh, they're just talking.
Oh.
Yeah, they were just having a discussion too. I think they were having a discussion about what you were talking about.
Oh. You could join. Do you want me to join or no? No?
We turned the talk function off because they said it has feedback problems back there. So we would put turn the talk function off. Now you can hear me because I just turned it back on. But I was asking, I think you're using different dyes. I was just asking why those different dyes, they are dying. I guess they label different places.
Oh, the cells? The size of the cells?
Yeah, yeah.
We have only, here we don't have any. Let's see. The size of this cell, the one that is infected, most likely is what happened to this? I did something wrong.
You have to put the eraser back. It remembers the last thing you picked up.
At least you didn't tell me.
No.
With the eraser. This cell is larger. It looks larger because it is infected and they start producing, they go big. Okay, and all these are just the nuclei of the cells, okay? The influx of cells into this particular cuff is mainly monocytes and T3 cells that can be T3 and T8 or T4, okay? And those will have a smaller nuclei like that, yes, but the big ones are most likely to be monocytes. We could stain for the different lineages and the different kinds of macrophages. For macrophages in the perivascular space is my main work, in the sense that we are studying the reservoir, how mobile is the reservoir of virus that is in the brain coming into the rest of the organism. Okay, because in the therapy to kill the virus that produces AIDS, the drugs don't go into the brain. Then they have a privileged area where they could reproduce. Okay, for me it's very important. Then you tell me you want to know what are they and so on. I have to give you a lecture with many markers, but the size correlates very well with the differentiation from monocyte to a functional macrophage. Okay, and from the macrophages nowadays we classify it in 3 different ways. We are catching up, like with in immunology they have a CD3, CD4, CD8, and CD25, and all these, many, okay, cluster designation numbers, okay. With macrophages, it's not that clear, and we are defining at least the ones that are involved in the inflammatory process in the brain, okay. Good. This is another good picture of a cell that is infected. This is with the virus. We generate a virus that has in its genome green fluorescent protein. Then we inoculate the animal, and then it gets AIDS, and it gets a virus that fluoresces, okay, in green. And there's these cells, okay. And what we found out, we were not looking at, for that, but the CD3, the T3 receptor is introduced into the cell that is infected. When they are not infected, like this one, it's all outside, okay? You could see this, okay? It's all outside. But when it's infected, all the red is inside, okay? With this one, nothing in the membrane. Well, when you say, oh, but that's too late, it's already infected. But it's part of the cellular response that is happening to them. Okay, and this is the kind of pictures that you could generate for your work, and then it illustrates very well many things. And I tell everybody that wants to work and that collaborates and so on, I tell people, aim for the cover of the journal. Okay. If you aim for the cover of the journal, any picture that you get will be published. Okay, this is something. and I am self-promoting. We have 13 cover pages in different journals, 2 of them with Gosco Salas, with their group. Okay, I'm not lying. All right. This I put in as to show you the kind of resolution. Unfortunately, we are not going to, the movie we could see later on, but this is one cell that is synthesizing IL-6, okay? And you can see all these little things, the endosomes, okay? And all this is the Golgi, okay? By culturing the tissue with blockers that block secretion, we are able to stain for the cytokines that they are going to produce. And then we have this thing that we could follow the production of cytokines in the tissue and the cells that are coming into towards that cells that are synthesizing something, okay? As we don't have the movie. Okay, let's see. Next one.
It might be that blank slide. Remember a blank slide in your...
Okay. All right, then we don't have the movie. This is also a movie. It is not, but this I put in to show you the kind of resolution that you could have with confocal microscope. This is a single cell vessel, okay? It's a vessel, it's a capillary that is made of one cell. When we rotate it, you'll see the light of the cell. This is another single cell capillary, and we don't know what is this. It's part of the membrane of the vessel. Okay, it's in the brain, and you could see that with this, you have the membrane on one side and then the membrane of the other side. Okay. Hopefully you could see that there are 2 lines there, okay, and 2 lines here. We're able to see the membrane that is going like this outside. And I don't know what are they doing. They are vessels. They shouldn't have those things. But it's very clear that these little arms are in close contact or tangled with the astrocytes. This is the astrocytes, okay? If it's part of the scaffolding to maintain the vessel in one position, it's okay. But we have been working always that they are separated. They don't touch each other in that way. The astrocytes have footing, okay, but they don't grab the thing. With this, they say, oh, they are so narrow that okay, they could be grabbing each other, okay, because in between the vessel and the astrocytes, there is some virtual space, okay, the perivascular space, okay. Then coming back to this, I'll say, oh, we have lots of pictures. We always could generate numbers with confocal microscopy. All your pictures have associated values. And for example, just measuring the density of the vessel, you could have numbers and say, oh. You could have graphs, and you could convince people. Okay, and then if I could help you to be successful, please call me, and you could do. With the guys that are doing PhD, the only thing that I ask from them is publish. You're not publishing, okay, I don't want to work with you. So I have to justify my time too, okay? What is this? There. Showing off, most likely. We were able to do cytokine chemokine detection for anybody in tissue. Okay, we could do in situ CTL tetramer detection. That is something that is extremely sensitive, is very cumbersome, but is very important. What we could do is detecting the exact cell that is going to attack the cells that are infected with the virus, okay, in tissue. And it has given us some good papers with that. With the colors, detection of different antigens and some, yes, we could go up to 6, okay. When the principal investigator needs something very specific and is not in the market, we are able to produce antibodies. I send them always to do this from chickens, because that gives us another species that we could detect for multicolor. Okay, but it's possible. We could generate. They could generate the antibodies for almost everything. And I started doing some super paramagnetic iron particles that are fluorescent and that are magnetic and they have iron. That will be for another day. Okay, we have many covers of journals. We are okay. Then that was a movie, but you know. Okay. And I think that I finished that one. Do you want to see movies or you had enough? Hello?
They're a quiet bunch.
Yes.
Make sure to put the marker down. Put the marker down. Then it knows you're in mouse mode. There you go.
Okay.
Say no on that question.
Say no.
No.
Let's see. No. 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 Okay. Oh, we take one. Hey. Oh. Unfortunately, I didn't talk about this, but this is very. Can I do?
Well, loop, you have to press play. Loop, do loop and then press play.
And I could stop this?
Yeah, you can pause.
Oh, okay. This is something that that we found recently. Blue is IL-12, interleukin 12. And then we see cells that are producing interleukin 12. And red is CD123. CD123 is the receptor for IL-2. And this is at the point where the cells come into the lymph node. The cells that are all armed or they show a lot of the red stuff, they have IL-3 receptors in order to check the cells that are coming through, okay? This is the marker that we are using to detect. This is a marker of monocytes that we use, and you could see these cells go there, and these cells are tessellated. I don't know if you remember the word from mathematics, whenever they fit together perfectly, that are forming a very good, like a basket, okay? They are tessellated, all of them, then they don't allow to pass anyone without being checked. And the funny thing is that the ones that are producing the IL-6, the IL-12 are passing. But this is just an example of a confocal that you could see there is room for doing these things for years. I think that is a very exciting part. I like it a lot because I see the pictures, and then you could figure out whatever is happening and so on. Okay, but when had you seen? And these cells are part of the red cells are dendritic cells. Okay, we found that they are forming all this basket around the lymph node. Okay, and these are the elusive dendritic cells. It's a pain trying to stain for them, but you could see functional. Oh, how do I close this guy? No. Now it wants to. Oh, with that one. Okay. Let's see. Oh. Cancel. The problem that we are having is I use Macintosh. None of them. No.
Maybe in the future you shouldn't use toy computers.
No, it's oh, it's there. What is this? Oh, and there's always what it is. It's CCR5. CCR5 is the co-receptor of the co-receptor of the cells to bind HIV. Okay, is normally they bind to CD4 and one of the co-receptors. This is the co-receptor that is used the most in monkeys. Then this and the 1, 2, 3 is as for dendritic cells. This is the presence of the receptor in dendritic cells. And you say, oh, what's that? And then we could demonstrate that those cells get infected. Okay, that's infected with the virus of AIDS. And I think that's enough. Any question?
Well, I have a question for both presentations. If we're interested in doing some microscopy imaging and we plan to send our samples or have our students come to your facility, and how do we coordinate that? And is there any particular fee charged? And is that, sort of those details we're kind of planning to do?
So you would send me an email.
Okay.
And then I'll help you depending on what you need, what type of application, what kind of services you need. I can help link you with any one of because Javier has his center at the Tulane Primate Center. Pete's at the vet school. We have another facility at the vet school and one in life sciences here at LSU. And we can help you find the right facility for what your needs are.
Yeah.
Each facility has its own set of fees that any researchers have to pay. So LSU researchers have to pay, and so it would be whatever the rates are for that particular facility. And we can help you negotiate that as well, and help you sort out what the cost would be. And in some cases, the molecular cell biology core may be able to assist in some of those fees.
That would be great.
Okay.
What's your email address, Mark?
It's mboudr2@lsu.edu.
And what's your last name, please?
It's Boudreaux, Boudreaux.
Okay, thanks.
And my first name is Mark, M-A-R-C.
June, I'm going to send you an email.
Okay, thanks, John.
I have a question about the facilities down there.
That's mine. Oh, I need to practice to write with this.
Okay, go ahead with your question.
I actually was looking for a transmission electron microscopy machine. I don't know, do you guys have that down there at LSU, at any one of those facilities?
Yes, we do. And matter of fact, next Thursday, we're going to have another session. It's the second session for this microscopy training, in which we'll have the leaders of two other cores, one at the vet school and one at life sciences, that both have SEM and TEM capabilities. So if you're able to attend next week's, you'll get to hear from Matt Brown and Dr. Wu about their facilities and what they can offer as well.
It's next Thursday, same time?
Next Thursday, same time.
Same time, same place.
Same bad channel.
Typically, if I want to send samples down there for characterization, what is the turnaround time?
That's just dependent on the facility. I can help facilitate that for you, but it's going to depend on the workload at each facility, and and that varies from time to time. But I can get you in touch with the facilities, and you can ask that directly to them, because I'm not currently sure what their turnaround time is.
Okay. Xavier, I have a few quick questions for you. You mentioned the Alexis dye as the fourth generation dye, and I work with FITC. I'm currently working with FITC. So what advantage does this Alexis dye provide over the FITC kind of dye?
The FITC.
The fluorescent isocyanate. Is it just this Alexis dye, do they excite at a longer wavelength, or is that the advantage, main advantage?
Let me give you just want to write it?
Uh-huh. And I go right.
I saw a fluorescence spectrum of the Alexis dyes, but I'm not sure that x-axis is. I didn't quite see the wavelength.
No, it's same color.
Which, yeah, blue, red, and the other one's a highlighter, sorry, and black.
This is Alexa 488, and this is FITC. Okay, and then for the if you want only one color, never mind. FITC works well. FITC quenches faster, about 1,000 times faster than Alexa. But we could go with the quenching. Okay, but the spectra is very different. And then if you want another color here, with the Alexa, it's very easy to. Oh, I see. With the Alexa things, it's very easy to use the Alexa 568. Okay. With FITC, we will have to use something that is more towards there, like Alexa 594, and then the spectrum is like that here. Okay. Or Texas Red. Then you have the chance of doing more colors. Alexa quenches very slower than FITC. FITC is susceptible to pH changes, and then if you put anti-quenching, it's okay. But a lot of people or they prepare wrongly, so the anti-quenching, or they use something just PBS, and then it quenches faster. That's the advantage. In my hands, if it is before they come to me, I ask them if, do this with something that is not a FITC, and then we could image. If they don't have the antibody, I could send you the antibody, or you could come and stain with me. The secondary antibodies are relatively inexpensive because they last forever. Okay, we use very diluted, 1 to 1,000, then use very diluted. That's the reason. Molecular probes do not give me any money. and when you don't have any other option, that's okay. We use FITSI. Okay, but it's a lot more difficult to put more colors.
Thank you.
Are there any other questions?
Oh.
All right. Thank you, Javier.
Thank you.